Oke, minggu harap.
Kabur boleh buat.
Minggu depan ke masuk nih.
Ngisi. Dikaburu libur ah.
Nah. Kudu nama minggu kemarin kan Paroho. Paroho teh kan beda jam na. Teteh angkat dulu. Assalamualaikum.
Waalaikumsalam. Waalaikumsalam.
Assalamualaikum. Selamat pagi.
Waalaikumsalam
warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam.
Sehat Bu Eslin?
Sihat, alhamdulillah. Boleh nampak saya punya slide? Saya sudah share.
Oke.
Ya, nampak.
Ya, sudah Bu, sudah terlihat.
Okey.
Nampak.
Sudah.
Saya sudah ready lah. Sini kita balik analitik ni kita buat contoh-contoh kan? Ya. Contoh ni lah. So kita masih menunggu partisipan?
Iya.
Oh, okey.
Mati lagi. Kita coba masih nunggu yang lainnya Bu ya. Bentar.
Boleh Pak, boleh. Tak ada masalah.
Assalamualaikum.
Oh iya.
Teman saya yang belum di esif Pak untuk masuk ke room Zoom-nya.
Oke, siap.
Ya, terima kasih Pak.
Ini ntar moderator sama Bu Winda, Bu Esatul ya. Bu Winda.
Okey.
Ya, salam kenal Ibu.
Ya, salam kenal Bu.
Sehat Bu?
Sihat, alhamdulillah.
Alhamdulillah.
Kalau saya tengok ni sangat kan. Kalau kita buat macam ni. Tekan. Kan? Itu dia buat ni juga apa? Kena dua-dua.
Lagi next sini, ini lupa. Sini dah bukan next, saya slide. Ini bukan mouse ya.
Macam next ni macam mana? Kita kat tengah ni.
Iya.
Mohon izin ambil gambar. So kita ada 790 participants.
Memang. Oh.
Sudah perkenalkan, jawabannya ada. Kuliah umum, ujian diganti kuliah umum ya, tapi tetap ujian gak jam 4.
malahan dia mau menampar mimpi, namun umumnya sangat prematur langsung ditahan. Kalau kalian pikiran adalah melakukan, kalian salah guys. Yang ternyata adalah ini, sedangkan Joseph malu mencoba bermimpi, ketakutan seperti ayam sayur. Ini tanpa takut langsung menampar preman itu. Dan bersama lihat, ini menyebut di rumahnya karena dia tidak mau mimpi terlambat lagi ke sekolah. Namun tanpa diduga, motor mereka dilanggar oleh anggota preman tadi menggunakan sebuah truk. Dan Joseph langsung panik, tapi untungnya begitu berkurang tidak kenapa-napa. Penny yang tidak terima dengan semua ini, kemudian tanpa semangat Joseph membuat laporan ke polisi. Sebenarnya belum ada yang sudah
Kita masih menanti ya Pak.
Bu, kita mulai aja ya Bu ya.
Saya tak tahu, terpulang kepada Ibu lah. Saya terserah.
Ya, kita mulai saja karena waktunya sudah 9.45 ya Bu ya. Biar nanti nggak telat. Oke, bismillahirrahmanirrahim. Ladies and gentlemen, attention please. We will start this guest lecture. The Honorable Dr. Ezatul Azlan from University Science Malaysia, Dr. Apoteker Saiful Amin as Vice Chairman in Academic Field of Stikes Bakti Tunas Husada Tasikmalaya, and Apoteker Ira Rahmiani, MSc as Head of Pharmacy Study Program in Stikes Bakti Tunas Husada Tasikmalaya, and also for all participants in this meeting. Good morning everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay. First of all, I'd like to welcome you all to this guest lecture which held by Pharmacy Study Program of Stikes Bakti Tunas Husada Tasikmalaya, collaborated with University Science Malaysia, Universitas Buana Perjuangan Karawang, Universitas Garut, and also Universitas Perjuangan Tasikmalaya. This guest lecture has a theme. The theme is Principles of Medicinal Chemistry. And before the presentation begin, allow me to introduce the speaker for our guest lecture today. Maybe I will share your CV, Mrs. Ezatul.
Ya, mungkin.
Okay. This is CV from Mrs. Ezatul Azlan. Her full name is Ezatul Azlan binti Kamarul Zaman. She is senior lecturer at School of Pharmaceutical Sciences, University Science Malaysia in Penang, Malaysia, especially in pharmaceutical chemistry. She was born on 29 November 1979 in Perak, Malaysia, and she got a bachelor degree in applied science, industrial chemistry in USM 2002, and then she got master science in pharmacy in USM 2006, and then she got a PhD in pharmaceutical chemistry, joint supervision between USM and Lorraine University, Nancy, France, at 2012. And then, okay. This is the research interest of Dr. Ezatul Azlan. The research interest is drug design and discovery of potential natural product, peptides, and synthesized organic compound for antiviral, anti-cancer, and anti-obesity properties. And then area of expertise, computational chemistry, medicinal chemistry, peptide synthesis, and small molecule synthesis, natural product chemistry. And she has a lot of research grant project. As you see at the screen, it's so many. And then, and also her publication in patent, international journal, and also in the local journal, so many experience. I cannot read one by one because it's so many, take a long time. So you can see at your screen. And then She also she is a reviewer also in journal. She has a book chapter, and then she is supervisor of postgraduate and also undergraduate student in University Science Malaysia. So many experience of her. You can see at your screen, and then she got many academic awards. You can see at your screen. So many, so I cannot read one by one. CP for Dr. Ezatul Azlan. Maybe for the next, I will please welcome to our guest lecture, Dr. Ezatul Azlan. The time is yours.
Thank you, Dr. Winda Krishna, for the pleasant introductory of myself. So, Assalamualaikum and very good morning to all. I would like to welcome participants, undergraduate student or postgraduate student, and also the lecturers from STI Kes Bakti Tunas Pusada Tasikmalaya, Universitas Garut, Universitas Perjuangan Tasikmalaya, and also from Universitas Buana Perjuangan Karawang. Thank you for your for inviting me today to give a lecture on the principles of medicinal chemistry. So I will elaborate the principles of medicinal chemistry in terms of drug discovery and development process. So before we start, we need to know what is medicinal chemistry. So medicinal chemistry is best to be defined as an interdisciplinary research area that incorporating different branches of chemistry and biology in the research for better and new drugs, the drug discovery. Or in other words, MedChem is the science which deals with the discovery and design of a new and better therapeutic chemicals into new medicine drugs. So generally, medicinal chemists can make a new compound. We can determine their effect on biological processes. We can alter the structure or we can modify the structure of the compound for optimum effect and to minimum the side effect. So we can also study the uptake, the distribution, the metabolism, and the excretion of the drugs. So in medicinal chemistry, the chemist attempts to design and synthesize a medicine or pharmaceutical agent which will benefit to human. So such a compound, we call it a drug. So in this process, it will involve drug design. We will synthesize the drug that we have designed. We will involve the structure-activity relationship, the receptor interaction, and also the absorption, distribution, metabolism, and excretion of the drug. So I will go one by one from basic, yeah. So the medicine is a drugs or pharmaceutics or any media distinction or any chemical or biological substance, either synthetic or isolate or non-synthetic, that can give the effect to the process of the mind or body. So the drug, this compound can activate or inhibit the function of a molecule such as protein, which in turn results in therapeutic benefit to the patient. Am I too fast? Okay, can follow?
Yes.
So before we start, we need to classify the drugs. First, we can classify the drugs in terms of biological or pharmacological effects. For example, we have analgesic, anti-asthmatic, antipsychotics, antihypertensive. So all this drug is useful in one wishes to know the full scope of drugs that are available for a certain ailment. But it means that the drugs included are numerous and highly varied in their structure. So there are a large variety and targets which drug could be act in order to produce the desired effect, or either to stop the disease, for example. So we can also classify our drug by chemical structure. There are too many drugs which have a common skeleton or group together. We can group them into a penicillin, for example, opiates, barbiturate, or steroid. Okay. We can also classify a drug by target system. For example, for antihistamine, they will only affect a certain target system in the body. Another example, the target system is where a neurotransmitter is synthesized. So it will release from its neuron that interact with a protein target and is either metabolized or reabsorbed into the neuron. So for this target system, this classification is a bit more specific. And the other target, which is more specific, which is by site of actions. So for example, we have anti-cholinesterase drug. So they will specifically bind or inhibit the acetylcholinesterase enzyme in CNS. So we can also, based on this drug classification, we can naming the drugs and medicine based on the availability or function, for example. So let's say the compounds that are synthesized in medicinal chemistry research, but never make it into the market. So it is impractical to name them. Normally people will just label the drug with a code, which consists of letters and numbers. For example, RO31 8959 for Roche. So from Merck, MK639, this is for example, we just it's not be able to be available in the market, just label with the code. If the compound concern show promise as therapeutic drugs, so they are taken into development and name it. For example, some compounds that promise as anti-HIV drugs, we ritonavir or indinavir. So the drugs that has been proved successful and are ready to be marketed in the to be sold. So normally they are given a proprietary brand or trade name. For example, just now I said about seconavir, ritonavir, and indinavir. So we market them. People market them as Photovase, Norvir, or Crizivan. Okay. So when a drug's patent, every drug normally has a patent file. So the pharmaceutical company will keep the secret of the drugs in order to protect their finding, their selling price, etc. So when their patent expired, now it is time or possible for any other pharmaceutical company to produce or sell that drug as a generic medicines. So actually, normally they are not allowed to use the trade name use, but they need to use the the generic medicine. For example, in the law, European law, it requires that generic medicine are given a recommended international non-proprietary name. So how drugs are discovered? Many times ago, the study or practice for thousands of years, people will use a root, plants, tree, berries of herbs in medical folklore in order to cure cough, for example, or in order to treat fever or to reduce temperature when people get high temperature or fever. Okay, in the last 115 years, the isolation and purification of the active principles of the remedies called the mechanism of reaction have been used. So in 1990, the first chemotherapy has been introduced. That it means that the chemical that directly interfere with the proliferation of the microorganism at concentration tolerated by the host selective toxicity. Okay, these are the opium that we obtain for morphine, heroin, or codeine. So when medicinal chemists would like to synthesize or would like to obtain a new drug, what should they think first? What are new? Why we need new drugs? Because we need to improve the medical needs. So maybe some of the drugs are not give a full activity, for example. So why we need new drugs? Because now we have new disease. For example, we have COVID-19. So now people in the pharmaceutical industry will work on the vaccine or on the to treat COVID-19. So why we need new drugs? Because some disease still with no treatment. For example, AIDS or dengue. If people got dengue, they will have fever. We just need transmission of the blood, and also if we have a fever, doctor just give us the paracetamol. So we need also new drugs because some of the drug will give a low efficacy. For example, dementia, cancer, or obesity. Some drugs will still give a side effect to a certain people. So why we need new drugs? We need to cut the cost of therapy. For example, in the case of cancer treatment, the cost to individual or to the country, for example, there's Alzheimer, spinal injury, and depression. So lastly, we need the new drugs for certain pharmaceutical company to sustain industrial activity. Okay. So these are top 10 the world biggest pharmaceutical companies that gain the high revenue in 2020. So normally the global pharmaceutical industry is expected to the weakness of the positive growth as the top pharma companies before the existing of the new disease. For example, the COVID-19, the close example in this case. So these pharmaceutical companies will invest in the study of the development of the COVID-19 vaccine so that people will get there are higher demand nowadays to obtain the COVID-19 vaccine, so people will buy. So they will sell it and they will obtain the revenue. So now I will enter to bring you all to the drug discovery process. So drug discovery is the process whereby a drug candidate or lead compound is identified and validated for the treatment of a specific disease. Okay, the process will go through with the potential of new medicines which are identified and involved in a wide range of scientific disciplines including biology, chemistry, pharmacology, etc. So after we make the discovery process of the drug, then we need to go to the development process. So in this case, we will bring a new pharmaceutical drug to the market once a lead compound has been identified through the process of drug discovery. So in this case, if you can see here, the drug discovery will be involved the target, the hit identification, hits to lead, hits to lead and hits optimization. So first of all, for example, we have we have 1,000 of compound, and then we go to hit to lead of compound with 100 compound, and then for the lead optimization we just have about 10 to dozen of compound in order to have a good, very good compound candidate that we seek for the preclinical development that will be involved phase 1, phase 2 and phase 3 of clinical study. Okay. Oh, what happened to my, how to, so we can undo. Oh, clear all drawing. Okay. Sorry, okay. First, when we want to make a new drug, we need to have in our mind what disease we would like to study. Okay. So nowadays, for the popular one is the COVID-19. Okay. So the pharmaceutical companies normally will target the disease that can have a commercial enterprise. Therefore, they tend to avoid drugs with a small market. So now people will study on and discover and develop of the COVID-19 because it has a high demand. So another things how to choose disease. The disease, most research is carried out on the disease which afflict the first world countries. The popular disease, for example, cancer, cardiovascular, depression, diabetes, flu, or obesity. Because when we have a lot of disease cases in certain country, we can easily sell the drug. So lastly, the most important thing, the pharmaceutical companies will also focus on the popular and up-to-date disease. The nearest example now is the COVID-19 disease. Okay. So far, any question? Any question from participant?
For all participants who has a question, please
I
raise your
think you
hand.
finish first, Bu S1.
Oh, at the end. So I have a question. Get in your mind how to choose a target disease. When you become or you want to further study to do your master or PhD or any research area, what disease that you plan to study. For example, in my case, okay, Malaysia has a lot of cases, dengue problem. So if I choose or write a proposal grant on dengue, I can, inshallah, will get it. So that is how we need to strategize what disease that we want to study. Okay, now we go to heat identification. So heat identification is how we identify our drug target. The drug target is a specific molecule or biological system which the drug will interact with. For example, the enzyme, the protein, the nucleic acid, the protease, all are the drug target. Okay. We can identify a drug target by using bioassay study. Bioassay is a test to determine the biological activity. The most popular one, the in vitro study. In vitro is an artificial environment when we have the protein, when we have the enzyme inside the plate, then we put our compound. So we can see whether they have the activity or not for that particular disease. So normally the bioassay should be simple, quick, and relevant. So for example, the enzyme we can just buy, okay? If we cannot buy the enzyme or protease, can we express and purify the protease? Do we have the plasmid to make the protein expression? Do we have the facility to make it? Do we have the money or human resource to make the protease? So normally for in vitro bioassay, we have it has advantage in terms of speed, and it requires a small amount of the compound to be tested. But if we have a lot of amount of compound, we can speed up our activity by using high throughput screening. I will explain later in the next slide. So drug target can also be identified in vivo using the living body. Normally we use animal, but normally we will not using animal test in the beginning of drug discovery because it is more expensive and it may cause suffering to animals. So we can also use ex vivo, which is usually refers to the test on a tissue that taken from a living organism. Okay, when we do the bioassay, we need also to make the test validity of our testing procedure. It should be easy and clear cut. For example, when we want to develop an antibacterial agent, so whether to test our antibacterial agent is good or not, we can test by measuring how effectively our antibacterial agent we kills the bacterial cells. So obviously, if they can kill the bacterial cells, then our antibacterial is a good agent. If not, vice versa. Okay, so we can also identify using a potential inhibitor, which can be first screen its percentage of inhibition. For example, here the figure on our left, we have tested the same compound with different concentration, 3 micromolar, 15, 60, and 120. So it will give a general idea with which concentration or which drug is a good on that particular disease. So once we have the rough idea which compound can give a high percentage of inhibition, we can proceed our test with IC50 value. With this, at 50% of inhibition, we can measure the concentration, the best concentration of the drug, our potential inhibitor. To obtain the IC50 value, we need to plot at least 5 to 10 different concentration of the drugs. Okay, we can also identify our target by using high throughput screening. High throughput screening is we need a leverage automation of machine or robotic machine that will run the assay for us. So when we just set up everything in the computer, we set up in the plate, so the machine, the robotic machine, will take the sample in the sample container and test the activity on each plate. Each plate can be the same protein or it might be different protein. Okay. We can also identify our drug target using NMR, nuclear magnetic resonance, which is a technique that exploits the magnetic properties of certain atomic nuclei to determine the physical and chemical properties of atoms or the molecules in which they are contained. In this case, we have to dilute to prepare our protein and ligand in NMR solvent. For example, deuterated solvent, for example, diameso, or deuterated chloroform. And then the tube will be placed at the top of the NMR machine here. Right here, okay. So we will run the experiment. It will give us the intensity in Fourier transform. So from here we can obtain the peak, and it will give us the electron density map, and we can guess the structure and the interaction of our ligand and with the protein. For example, here we have a functional chemical library of our ligand that we mix with our protein and we run the NMR. We get the proton and 2D proton NMR of the compounds to confirm the specific binding, and we can also identify the active site. So with the active site, we can map on the protein structure. Then we can see we can then run the autodoc structure of the protein ligand complex, and we can identify the similar binding site of the protein and the computational approach. Oops. So we can also identify our drug target using X-ray crystallography. In this case, the ligand, the compound, the drug, will be together with the protein need to be crystallized. So when we obtain the crystal, it's very small crystal structure. So we put the crystal structure here at the goniometer. So I use the other chip. Okay. So we place at the crystal here. Okay, and then the electron beams, electron beam will bend the crystal and pass through the crystal and to give us a diffraction pattern. From this diffraction pattern, we need to use a software that can identify the electron density map that later produce the atomic model of our protein and ligand target. So I'm clear. Okay. Okay. All the data that people publish in NMR and X-ray crystallography data are deposited at rcsb.org protein data bank. For example, here, hopefully we can enter to this website. Oh, never mind. I already clicked, but it doesn't work. So you can go to the protein data bank and search any protein target that you want to study. Before you want to choose that protein target, you must read a lot, make a literature review. If you type COVID-19, there are hundreds or thousands of COVID-19 protein target. So which one you want to use, which one you want to choose. So that is the importance of reading. You need to make a thorough literature review, because when you make a wrong decision at the beginning to choose the protein target, you will lose it at the end of your project or study. So normally in protein data bank, we will have a file, PDB file, something like this. The atom, the number of atom, the name, nitrogen, hydrogen that connect to nitrogen, carbon, aromatic, for example. So the residue name, alanine, because alanine, serine, threonine, tyrosine, methionine, histidine, because this residue name come from our the amino acid. Our protein is linked from a certain amino acid, 100 to 1000 or whatsoever amino acid, in order to obtain one protein or one protease, for example. So we have a change. Sometimes when we people do the X-ray crystallography, they will obtain change A, B, C, D, whatsoever. So we have the residue name, residue ID, residue 1 is alanine, residue 3 is alanine, and residue 4 is also alanine. If we have residue 4 is threonine or tyrosine, the code here TYR, for example. So then we have X, Y, Z coordinates of the particular protein. So then we have a 3D model of the protein. So there are another approach to get a drug target, to know the drug target. So this is quite a new technique. We call it isothermal titration calorimetry, or ITC. So ITC is a technique that is used to determine the thermodynamic properties of binding between a drug and its protein target. How it can be useful in order to see the drug, to get the drug target. Let's say we have a ligand. Either we synthesize in the lab or we isolate from a plant. We put it our ligand here. So in the chamber here, in the downside chamber here, we already have our receptor, which is our protein, protease, nucleic acid, enzyme, etc. So when the ligand is dropped into the receptor, we will use a heat. And with the droplet, we use a titration procedure or method. So when they first drop, they will plot a graph here until we have at the final titration, we will have a plateau graph. From this graph, we can calculate the thermodynamic properties of the interaction between the ligand and the target. In this case, we will obtain the dissociation constant of the interaction, the molar free energy change, delta G, enthalpy change, delta H, entropy change, delta S, and also the stoichiometry of the protein ligand binding interaction. So if the delta G value is low, we know that maybe we have a negative value, we know that it has a good binding interaction between a protein and ligand. If the value is high, we know that that one is not a good interaction of protein ligand binding. Okay. So far, any question? So we need to recap. So how to identify a drug target? There are at least 8 techniques or approaches to identify a drug target. In vitro, in vivo, ex vivo, test validity, high throughput screening, NMR, X-ray crystallography, and also ITC. So now we will, excuse me, in some water. Now we will enter to the heat to lead process. We already know how to obtain our protein. We already know how to choose a target disease. After we choose disease, we need to know which protein that we want to study. So now is the process to find a lead compound, which drugs are good for our particular enzyme or macromolecule. So first of all, we can screen, we can find the lead compound from the screening of the synthetic banks. Many pharmaceutical companies or some researchers have already prepared thousands of compounds in their library. So they just store in the freezer, make catalog, and then they just take their compound in the library and screen on a new target as these new targets are already identified. So, and then the second strategy, we can get the lead compound from screening natural product. There are many natural products that has been isolated and identified for a certain particular disease. For example, this one, we obtain this compound, pancreatistatin, from Pancratium littoralis, which is the beach spider lily that is useful for anti-cancer drug. These are another example of screening from natural product, which is obtained from marine invertebrate Bugula neritina, which is biostatin 1, that is also useful in the now in the under clinical development as anti-leukemic agent, and also in phase 2 clinical trials against melanomas, lymphomas, and renal cancer. Another strategy to find a lead compound, we can use it from the natural ligand or modulator. So we can use natural ligand for receptor. So normally the natural ligand is of a target receptor has sometimes been used as the lead compound. For example, the adrenaline and noradrenaline were the starting points for the development of adrenergic beta agonists, such as it can come up with the drug salbutamol, dobutamine, and zamoterol. Oops. So we can also find the lead compound from the natural substrate for the enzyme. For example, it can be used as the lead compound in the design of an enzyme inhibitor. For example, in this case, enkephalins, which has been used as a lead compound for the design of enkephalinase inhibitors. So these are another strategy to find a lead compound. It can be also natural ligands as a lead compound that we discover from the cannabinoid receptor in the early 1990s, which lead to the discovery of two endogenous cannabinoid messenger, the anandamide and also the 2-arachidonyl glycerol. So such agents may prove useful in the suppressing nausea during the chemotherapy or in stimulating appetite in patients with AIDS. Oops. We can also find our lead compound when we modify a structure of the compound from the existing drug. For example, we can design the structure which is similar to the existing lead, but different enough to avoid the pattern restriction. In this case, one of the example when we modify the existing drug to improve the side effect. In this case, we have sulfanilamide. So sulfanilamide is an antibacterial drug with the side effect to lower the glucose level in the blood and also diuretic activity. However, at this part, when we change it into functional group to this one, so from sulfanilamide, we can obtain tolbutamide, which is useful in the treatment of type 2 diabetes. This is how we use this strategy in order to improve the side effect of the existing drug. So I need to do, okay, all drawing. Oops. Okay. And then, the fifth strategy to find a lead compound is combinatorial and parallel synthesis. So this is a automated solid phase procedure that aim to produce as many different structures as possible in a short a time as possible. So we will produce a mixture of compound in each reaction vials by generating the compound libraries from molecular building blocks. For example, let's say we have molecular building block that we color it with yellow color that combine with the green one and green two, and we have the blue one, blue two, blue three, and another pink and orange. So in this combinatorial or combinatorial synthesis, we combine each building block with another building block in order to obtain several other molecules. Okay. So from 9 building blocks, for example, we can obtain another 27 molecules. So this molecule we can use then as an in vitro assay in order to see whether this compound is good or not for that particular disease. So for parallel synthesis, for example, we have when we synthesis A plus B, we obtain AB, general parallel synthesis. Yeah. In this case, we will combine the parallel and combinatorial chemistry or synthesis in order when we have A plus B, we have A1, A2, AN, whatsoever number. We combine, we synthesis using parallel A to B1, or combinatorial A1 to B2, A1 to Bn, A1 to B3, for example. Then we will obtain a lot more of compounds. So these are the combinatorial and parallel synthesis strategy. So the sixth another strategy to find a lead compound, we can use computer-aided drug design or CADD. So CADD can help us CADD is the help of computational program to search for a lead compound. We can use if we know the precise molecular structure of the target protein, we can obtain the 3D structure from the protein data bank, for example. Then we can use computer to design a perfectly ligand that can fit at the binding site of that enzyme or protein. So these are several computational application of molecular modeling package. We have molecular docking, molecular dynamics simulation, pharmacophore, virtual screening, homology modeling, and de novo drug design. So you need to know which approach or which technique of molecular modeling you want to use. So we can also identify our lead compound using a structural database. We can use computerized searching of this particular database. So normally we will use a virtual screening that normally involve the use of computer programs to assess whether known compounds are likely to be lead compounds for a particular target. So in this case, all these compounds, 1,000 of compounds can be obtained from PubChem database, for example, zinc database or NADI database. NADI database belongs to our Dean, Prof. Habibah Wahab. She developed NADI database many years ago. In NADI database, we have a compound from Malaysian plant. So as you know, Malaysian plant is not much different with Indonesian plant. So if you're interested to use our NADI database in order to make a screen in your particular disease or protein target, you can contact Prof. Habibah later. Okay, now some of the compound of the lead compound can be found by a chance or serendipity. For example, when we want to study compound A for dengue, for example, luckily this compound A is not meant for dengue. It's meant for influenza, or it's meant for COVID, for example. So that is the one of the technique which is based on luck. So for example, the discovery of penicillin or development of Viagra to treat the erectile dysfunction. So this based on the serendipity, by chance that occurred at that time, by luck. I can say by luck. Okay, another approach to find a lead compound is based on the ethnobotanical approach. Ethnobotanical approach is the traditional use of natural organism for medicines. So normally we will use the systematic screening. Okay, let's say our grandfather, grandmother has used plant A, for example, hibiscus, to reduce the fever, for example. So we can take this opportunity to get the hibiscus plant in order to study whether it can release the reduce the temperature or not. So we can also, for example, plant B is normally our great grandfather will use to treat cough. So we can study the plant based on the traditional uses by our oldest, by our seniors, or by our grandfather, grandmothers, etc. This is the strategy, one of the strategy to find a lead compound. So a question, so you need to bear in your mind how to find a lead compound. So it can based on the generally with these 9 strategies. The screening synthetic bank, screening natural products, which is starting from the natural ligand or modulator, modified from the existing drug, make it as a combinatorial and parallel synthesis, computer-aided drug design, computerized searching of structural database, which is virtual screening, by chance, and also the ethnobotanical approach. So now we go to, excuse me, I want to drink some water. Okay, and then once we already discover the protein, and then we try to find a several lead compound that has a potential activity with our protein target, then we need undergo to the lead optimization. In this lead optimization, we have a lot of our ligand, our protein, our compound, potential compound. So which compound is really good? Which compound we can bring it into the market? All of them? It's not possible because some of it may be non-active, some of it maybe has the toxicity, some of it maybe has less absorption. So with all these compounds, first of all, we need to get which compound will be active or which compound will be non-active to our target. So once a lead compound has been discovered, it is important to understand which structural features are responsible in its biological activity. So again, we can do the bioassay, and we can, in this case, we can identify, let's say in this case, compound 1, 2, 6, 7, 10, 11, 9, and 19 are active in our particular target. So we will just focus on this compound. We just study the structural features, the functional group of this compound to be studied. Okay, because if for the compound, non-active compound, no point we study because it will be not a good compound later on. Okay, before we move to that particular optimization, we need to recognize the type of interaction that occur between our ligand, in this case the pink one here, and our protein. Either they make a hydrogen bond interaction, or van der Waals interaction, or hydrophobic interaction, or pi-pi stacking, for example. When we recognize the type of protein, we need to know the binding roles of the certain functional groups. Okay, our drug must have a lot of functional group. For example, alcohol, phenols, amides, carboxylic acid, ester, aldehyde, etc. We need to know the binding role of alcohol and phenols generally at the binding site of the protein. These two functional groups, alcohol and phenols, are commonly present in drugs, often involved in hydrogen bonding. So the hydrogen can act as a hydrogen bond acceptor, and the hydrogen can act as a hydrogen bond donor. So for example, in this case, we have, first of all, you need to know what is hydrogen bond. Hydrogen bond is an interaction of an electron of electronegative atom that are bonded to the hydrogen that make an interaction to the hydrogen that bonded to another electron negative atom. So what is electronegative atom? In this case, oxygen, nitrogen, or sulfur. So in this case, we know which one will. This one is the hydrogen that bonded to the oxygen here. So this is our oxygen, electronegative atom. This hydrogen that bonded to the electronegative atom will act as a hydrogen bond donor. So the other hydrogen from the binding site, from our amino acid or from our protein, will act as the hydrogen bond acceptor. The oxygen from the amino acids. Okay. So I clear. Okay. So in our ligand, we also have aromatic rings. Of course, in the protein also we have the aromatic rings which come from the, certain amino acid will have aromatic rings. So normally aromatic rings are planar, hydrophobic structures that we commonly involve in van der Waals interaction with flat hydrophobic regions of the binding sites. For example, in this is aromatic ring. This is aromatic ring from our ligand. Will make a hydrophobic interaction with the hydrophobic binding region of our amino acid. We have commonly you have 20 types of amino acids. These are some of the amino acid has hydrophobic properties, non-polar, polar, etc. So for hydrophobic interaction, they will make the interaction with between the aromatic rings and the aromatic from the or hydrophobic binding region from non-polar amino acids. So in our ligand, we also have alkenes. Like aromatic rings, alkenes are planar and hydrophobic. So alkene can interact with hydrophobic regions of the binding site through van der Waals interactions. So alkene is the carbon double bond, carbon double bond. Okay. So the properties is planar and hydrophobic. So they will make a hydrophobic interaction through van der Waals interaction. These are the binding comparison of an alkene with an alkane. When we have double bond here, and also we have a single bond C to C. For alkene, C double bond C, it will make a hydrophobic or van der Waals interaction from the carbon, planar carbon, to the hydrophobic binding region. For alkane, the single bond, it will make the hydrophobic region between the hydrophobic region and also the hydrogen atom. So then we need also to recognize the binding role of ketones and aldehydes. Ketone and a ketone group is not uncommon in many of the structure studies in medicinal chemistry, but it is important. So ketone is a planar group that can interact with the binding site to through hydrogen bonding, where the carbonyl oxygen acts as a hydrogen bond acceptor. And the other one is aldehydes, which are less common in drugs because they are more reactive and are susceptible to metabolic oxidation to carboxylic acid. However, aldehyde could interact in the same way as ketone. Okay, so they will make a hydrophobic region that are possible for the carbonyl group. And they also make the hydrogen bond interaction with the binding region through the carbonyl planar group. So we need also to know the binding roles of amine when we want to design a new drug. Let's say that particular drug should have a hydrophobic activity with the binding residue. So which functional group that we want to replace or we want to substitute at that particular functional group. So this is why we need to know the binding role of amino acid. Which functional group will give the hydrophobic effect or hydrophobic interaction with the ligand, with the binding site. Which one will give both hydrophobic and hydrogen bond interaction? which one will give only hydrogen bond interaction. So in another example, we need to know the binding role of amines. Amines, we will have primary amines, secondary amines, and tertiary amine. When we have primary amines, we will possible to have two hydrogen bond donor from the hydrogen that attach or bonded to the nitrogen. So for secondary amine, we will have only one hydrogen bond donor, and for tertiary amine, we don't have the hydrogen bond donor because they don't have hydrogen that bonded to the nitrogen. So for amines, it's possible to make ionic bond interaction because when the nitrogen will have a positive charge, they will make interaction with negative charge of atom from the binding site. So now we will see the binding role of amides. So amides are likely to interact with the binding site through hydrogen bond. So the carbonyl oxygen atom can act as a hydrogen bond acceptor and has the potential to form two hydrogen bonds. So in this case, we also have the in this case the figure A, the interaction of the nitrogen lone pair with the neighboring carbonyl group in amides will produce the positive charge here to make the interaction with binding site. Lastly, I would like to, there are a lot of others binding site, ester, ether, etc. So I think I will today I will just mention about the binding role until binding role of carboxylic acid, the most common functional group that we have in our drug: alcohol, phenol, ketone, aldehyde, alkene, aromatic. Okay, so for the binding role of carboxylic acid, we have the carboxylic acid is reasonably common in drugs. It can act as a hydrogen bond acceptor, or it can also act as a hydrogen bond donor. Where we need to recognize which part will give you the hydrogen bond acceptor, hydrogen bond, weak hydrogen bond acceptor is at this part because they bonded with the hydrogen bond donor. Okay. We have also, lastly, I think this one is the last one, binding roles of esters. Esters group has the potential to interact with a binding site as hydrogen bond acceptor only because they don't have hydrogen that bonded to the electronegative atom. So they will just accept the will accept the hydrogen bond acceptor. They are hydrogen bond acceptor. They don't be a donor. So once we recognize all the function of how the binding role of ketone, alcohol, phenol, aldehyde, ester, carboxylic acid, aromatic, etc., so we can perform a structure activity relationship or SAR study. So SAR is the aim of SAR study, structure activity relationship study, is to identify those parts of the molecule that are important to biological activity and those are not. For example, let's say we have this type of molecule. At the green, at the black one here, we need to modify of this imidazobenzodiazepinone because the structure are not decisive for mutagenicity. So in this red one, ON arrangement of the oxadiazole ring will causes the mutagenicity even for non-allele side chain side chains. So the blue one here, the structure with allele function are positive irrespective of on or no arrangement of that particular molecule. So at this end of this part here, we can change it into dipropyl, 2-dibutyl, dibutyl in order to have the methyl side change negative with no arrangement of heterocycles. So in this green part here, the arrow here, if we longer the change, it will reduce the effect. So we need to maintain that particular long change. We don't need to add more change at that particular part. So this is another example whether this part, the carbonyl group is necessary or not, or at this part. So this is the SAR study. Whether the carboxylic acid part is useful during formulation or the drug development, we need to keep it or not. Whether it will disrupt the interaction of the hydrogen bond, which part will make a hydrogen bond, which part will make the hydrophobic, which part we need to maintain, which part we need to replace. So this is what SAR structure activity relationship study will do. So with this SAR, we can know the relationship between the chemical or 3D structure of a molecule and its biological activity. For example, for compound 1 and 2, if we change at that particular functional group, we still have good activity. They still can produce the activity. However, when we remove the molecule and also the O-methyl group at that aromatic ring, we put another methyl group at that side change, it will give us a non-active compound. So these are this analysis of SAR will enable us to determine of the chemical groups that responsible for evoking and target biological effect in the organism. So this modification will later affect or will give a potency of a bioactive compound by changing its chemical structure. So for SAR study, it can be useful for target potency, receptor to improve the receptor drug interaction, to modify the structure of compound in order to modulate the metabolism, or to improve the bioavailability in order to improve the physical properties and formulation of the compound. Let's say that is already good compound, but it cannot be absorbed in the body because it is too hydrophobic. So we need to find which part will give the hydrophobic properties. We need to remove that part and replace with more hydrophilic functional group. So with SAR study, we can also make target selectivity. We can modify to reduce the toxicity and also to allow the patentability. In lead optimization, we can also use, do I still have time? So in lead optimization, we can also use a pharmacophore. Pharmacophore is a technique to make a relative position of important binding groups in space and disregard the molecule skeleton that holds them. For example, let's say we have a compound named dopamine. We need to identify the pharmacophore ID of this particular drug. So this is the structure. So we need to identify the structure of the drug. So this center will give you the hydrophobic or hydrogen bond interaction. This one part of aromatic, amine, etc. So we put it the center, and then we remove the skeleton of the compound. We just leave it the pharmacophore ID of the compound. And then when we identify the pharmacophore triangles, we will obtain a triangle, pharmacophore triangle, something like this. So each triangles will give us different distance from this one to this one, but and also will give us different angle of each particular trajectories. Okay, so this pharmacophore ID or scaffold will be then used to identify any other functional or any other molecules from our database or our synthetic bank in order to find which other compound will have the same pharmacophore ID as dopamine. So let's say from 1,000 compound, 1,000 compound, 500 will have the same pharmacophore ID as dopamine. They have a hydrogen bond donor acceptor and aromatic at that particular position, so that we can focus to study this 500 compound for as a lead compound, or we can also to optimize this compound. So what will we do once the 3D pharmacophore has been identified? So the structure can be analyzed to see whether they can adopt a stable conformation which will contain the required pharmacophore. If they do, there are no steric clashes with the binding site, the structure should be active. We can use it for our further study. So another lead optimization technique or approach that we can use is ADME. The absorption, distribution, metabolism, excretion, and toxicity. Before we further study, we can test our compound whether it can be absorbed in the body or not. So to see the absorption, first we can check the solubility profile of the drug. We can calculate the log P of the compound. Once they have a good log P value, they can pass through the make a distribution to the body. So we can check their properties in plasma protein binding using experimental, and also we can use ADME, computational ADME, in order to predict the ADME properties of our drug. So we already finished with the discovery process, and now we will enter to development process. For development process, it will involve once we identify which compound is promising for the development, we need to conduct several experiments before to get some information how the drugs absorb, how the drug distributed, metabolized, and excreted from the body. Is it potential benefit and how about the potential benefit and mechanism of action? The best dosage, we need to calculate the IC50, the toxicity level of the drug. So we need to know what is the best way to give the drug at the oral or injection, what is the side effect, the toxicity, how it affect the different groups of the people differently, different race, different gender, different age, different country, country region, how it interacts with other drugs and treatment. If the patient already take another drug to treat for diabetes, for example, how it will affect the when people take another drug or have another treatment. So we will also need to know the effectiveness as compared with the similar drug. So before we go to the phase 1, phase 2, phase 3 clinical trials, we need to do the preclinical test. It will involve the animal testing to see the drug action, the behavior and the reaction in terms of physiology and histopathology of the drug, the toxicity, and also we need to consider the ethical aspect in order to carry out this in vivo test. We need to consider the number and a kind of animals that we use, either mouse, rat or rabbit, something like that. So then we can go to the preclinical development to check the pharmacokinetic and pharmacodynamics of the drugs. So what a body does to the drug in terms of pharmacokinetic studies, we need to investigate the liberation, absorption, distribution, metabolism, and excretion of the drug. And what a drug does to the body, we need to investigate the physiological effect, the drug action, and the relationship between drug concentration and effect. So in phase 1 clinical study, normally people will focus on the safety and pharmacology of a drug. People will start with the low dose first, and generally people use 20 to 100 volunteers. And then in phase 2 clinical trial, to see the effectiveness of the compound, so it will involve a larger number of patients, 100 to 300. And then during the, sorry, some water first. So, okay, in phase 3 clinical studies, they will involve researchers who would like to confirm the previous finding in a large number of population. So normally the phase 1 to 3 clinical trials will be involved many years. It's not an easy job to do, or it's not an easy task or short time to produce or to obtain one drug in the market. It has a long process from drug discovery and then drug development. So normally in drug discovery, we have thousands of compounds that we screen for the clinical, preclinical trials. For the clinical trials, we will normally have 5 compounds to be tested, and then only one particular drug normally will be approved by FDA, and we can market it in the or we can sell it in the world. So that's all my lecture for today. Thank you so much. Any question?
Yeah. Okay, thank you very much for Dr. Azla for the informative and interesting talk. For the next session is question and answer session. For all the participants who has question, you can raise hand and mention your name and your affiliation. Is there any question? Or I will read the question from the chat.
Okay. Can I see the chat? What is the chat?
Okay. I will share.
Oh, you will share. Okay, I will stop sharing my slide. Stop share.
Okay, okay.
Okay, I stop share my slide. Okay.
Okay. This is the question. Can you see my word?
Yes, there is a lot of
Dr.
questions.
Aizula?
So first, question number 1 from Asifa Aziz. Asifa Aziz from what university? Tasikmalaya Perjuangan, Tasikmalaya.
Check. Can you hear me?
Yeah.
Check, check.
So Asifa Aziz, first question, application of combinatorial and parallel synthesis chemistry methodologies to drug discovery. Can we do that? So how the synthesis parallel can be
Pak Rus? Suara saya ada Pak Rus?
Ulangi aja, Bu.
Saya dengar suara
Dengar.
Ibu Winda.
Dengar, Bu.
Cek, cek.
Slide-nya dibesarin aja, Bu Winda.
Oh iya, iya.
Ibu, saya bisa jawab.
Okay, okay.
Okay.
For the first question from Asifa Aziz.
From Asifa Aziz, the question is about the application of combinatorial and parallel synthesis chemistry methodologies to drug discovery. Can we do that? So how the synthesis parallel can be a new drug? If you change a different functional group, different molecule, it can be a new drug, no problem. So okay, so you just change okay, if the normal one, the functional group is hydrogen, you change to hydroxyl group or methyl, you will have a new drug, no problem. You just make a simple modification, or maybe you can choose with the you put the halogen atom, fluorine or etc. You will have a new drug. So Maulana Yusof, question number 2, should researchers of traditional medicine focus on development of new drug or validation of classical formulation? Thank you. Should resolve the you can still focus on the traditional medicine and development, but nowadays we have several computational approach in order to make your research more faster. For example, if you have 100 plant, so will you do the isolation extraction of the plant one by one, or you just predict the which plant that you want to study? For example, if I were you, I will try to find any chemical compounds that already published in that particular plant 1 until 100.
Yeah.
So then I will perform a reache screening. So in order to predict the interaction of this compound from that plant that can bind or can inhibit at the binding pocket of the protein or not. If they can bind, this is using computer first. If they can bind, I will let's say that one is the luteolin. As you know, luteolin or quercetin exists in many compounds, in many plants, sorry. So I just focus which plant will have most abundant of luteolin. Let's say plant A has a lot of luteolin in the fruit. Plant B has a lot of luteolin in the leaf. So I will choose the plant that I want to study that has a luteolin in the leaf, because leaf can be obtained a lot rather than to have luteolin from a fruit. Maybe that one is a seasonal fruit, so you need to wait the season of the fruit to obtain the luteolin. You get what I mean? Okay, Maulana Yusof. So can I go to the question number 3? So what standards allow phase 2 clinical vaccine to be administered for vaccine to be nowadays for the COVID? Hard to answer this question. Normally, phase 2 clinical or phase 1 clinical, we want to test for the effectiveness and the safetyness. So what happened if the vaccine has not reached the stage 3 by reason of an emergency situation? I have no idea to answer this. I afraid if I answer this, it will be people will ban me or whatsoever. So far the in this I think in this case the close situation that we have now is the COVID-19 vaccine lah, right? So yeah, many people will be argue about the safetyness, the effectiveness. So once I think once WHO has already approved the vaccine, inshallah we can use it, and that's all my comment. So question number 4, I want to ask the question about can the drug whose distribution permit has been withdrawn, its core structure be reused for the development of new drugs? I think yes. So the drug whose distribution permit has been withdrawn, its core structure be reused for the I think yes. It can be reused for the development of new drug with some improvement lah. You need to see is withdrawn based on what? Maybe based on it has a toxicity. So if we have a toxicity, we need to know which part will give a toxicity properties or value. So we can change that when once recognized. We apply our structured activity relationship study. We can change that particular functional group into another functional group. For example, the solbutamide and the tolbutamide just now, sulfanilamide and tolbutamide, it can improve the side effect of the sulfonamide. How can we make sure that our new lead compound has a potential activity to the specific disease, but all we know that everyone has different metabolism? Oh, you need to test. You need to test the assay to make the once you predict your lead compound, you need to test whether your lead compound is good or not by perform the in vitro activity. The in vitro activity, you have the protease, you have the you need to compare with the control drug control. For example, you want to test for influenza. So the protease is neuraminidase. So you can check whether your new drug is good or not by comparing your result with the approved drug, oseltamivir, the Tamiflu that normally used for the neuraminidase. So you can, by the comparison with the control, you can know whether your compound, your new lead compound has a potency or not to the specific disease. Okay, I think I answer. So with the issuance of an emergency use authorization of many COVID-19 vaccine from FDA, in a long run test, will the vaccine that get this authorization will be tested for further clinical testing if the COVID pandemic has ended? I have no idea to answer this one. Maybe or maybe not, I don't know. Maybe they are still in the clinical testing. That's all my comment for this hot topic to discuss about the COVID-19 vaccine.
Yes. Maybe for the next question.
So I move to the next question. How about the opportunities for traditional medicine, especially from plants that have potential to be a drug? They have same effectiveness as a chemical drug. As we know, every people has a different of metabolism, so that we can develop it for new drugs for current pandemic. For the current pandemic, the opportunities for traditional medicine, especially from plants, a lot of journal or publication nowadays that focus on COVID-19, they will make a computational work because they still need to develop the assay of the COVID. what people can do for the approach to treat COVID, approach or to suggest a potential drug for the COVID is the using computational work, just a prediction. Now I don't see any experimental data yet for this particular study lah on COVID-19. So next question from Dinda Putri. It was said that drug development follows current disease, whether new drugs will continue to be produced or not. If not, how will the company handle the cost of research? They will continue. The pharmaceutical companies will not stop their research. They will do the research every day, try to improve the effectiveness of the drug, try to improve the cost of to obtain the drug. So if it turns out that the drug doesn't meet sales target, so no, they will continue. If they stop the research, they will stop the to obtain the money. So for example, the synthesis of Tamiflu or Cetamivir, if you see the protocol to synthesis, it will need at least 17 steps to obtain oseltamivir. That is why people will continue to study how to obtain a better drug than oseltamivir in order to reduce the cost with the same effectiveness as oseltamivir, Tamiflu, for example. So any other question, Ibu Winda? The Dinda Putri is the last one.
There is some question in chat room. I will read for you. This is from Imas Tantri from Stikas Bagdetuna Sustada. She asked about NMR and X-ray crystallography. Between NMR or X-ray crystallography, which one that having the best result to describe or explain about new drugs? We can't just choose one or must do it all about NMR and X-ray crystallography. Thank you.
Oh, there are criteria. Which one we can use the best? For example, for NMR technique, it can only measure for a small ligand protein size, more or less about 100 residue only. But for depends on what is your protein. But if your protein has thousand of residue, normally people not able to use NMR to obtain the protein ligand interaction using NMR. So people will use X-ray crystallography. Okay, when people use X-ray crystallography, you will have 10 or 20, 100 files regarding that particular. So how to choose the best one? You can choose, you can see which one is will give you the good resolution of NMR, of X-ray crystallography, sorry. The high resolution that they use will capture the most residue or the most atom in the protein. Remember, when they want to get the protein ligand interaction using X-ray crystallography, they will make a crystal of the ligand and the protein. And when the X-ray beam pass through the X-ray, if the X-ray beam has a low resolution, they will miss a lot of information. Maybe some of the atoms are overlapping, they cannot capture, they cannot recognize. So the high resolution will capture a lot of, the high resolution of the X-ray beam will capture a lot of atoms. They will be not, they will have a less missing atom. So maybe for the low resolution of X-ray beam, we will have a lot of missing residue. Then missing residue is at the active site. So no point to choose that file as our protein target. So we need to, it's a lot of study, it's a lot of literature survey we need to make before we choose the right protein. Because when we choose the wrong one, everything will be wrong. Chang in, chang out. That is what Prof. Abba told me when I was a student with her. So another question, Ibu?
Yes, from Sylvia Tasha from Stickers BTH, we can find a lead compound with a natural ligand. Is there a natural ligand as the same effect to people? As we know that people with the same size and body composition can have different metabolic rates, especially with people with the different size. Thank you.
Yeah, when we want to find the lead compound, it's just the idea whether the compound will be inhibit or will be bind at the particular protein or target. So further study need to be done. That is why a lot of chemical candidate going to the phase 1, 2, 3 clinical trials, but sometimes not all will be passed through the clinical trials. Yes, because everyone has a different metabolomic profiles. So that is why when they go to the clinical trials, we will test on different people, different age, different gender, different race, etc. And different dose. I think that's all. I hope I answer your question.
Yeah. The last one, Dr. From Isprihatini from UBP Karawang. The question is how the chemical drugs react in the body. If there are side effects in the body, what to do first?
This stage of chemical react in the body during the phase 1, phase 2, phase 3 clinical trial, is it? Think so, yeah. So, if the drug react in the body, if there are some side effect, normally people okay. Normally when we do this type of I have no experience in this phase 1, phase 2, and phase 3 clinical trials. But when what I read and what I heard from my from certain people that have been doing that particular test, normally we will inject to the normal and also the patient. We will they will monitor after the injection what will happen with the first dose, for example. So if after maybe after 10 minutes when we have the side effect, let's say rashes, for example, or swelling at any part of our body, we need to tell the doctor or the person in charge immediately in order for them to know that our body doesn't accept that particular chemical entity, the new chemical entity. Just tell the doctor. I hope I answered the question.
Okay, Dr. Ezatu, that's all for the question. Maybe the other participant who has still the question.
Final one.
Okay, before if there is no question, before I close this meeting, maybe from Dr. Kaka from Unpar, is there anything to say? Or Mr. Saiful Amin from Stickers BTH or from Universitas Buana Perjuangan Karawang? Is there anything to say?
Or maybe later, if you still have some questions, can email to me, no problem.
Oh yeah. For all the participants who still has the question, you can send email to Dr. Ezatul Azlan.
Oh, I type my email here.
Yes, in chat room maybe.
Yes, in chat room. Okay, that is my email, izzatul@usm.my.
Okay, thank you, Dr. Ezatul.
Thank you so much, Ibu. Can we take a
Is there
photo
anything
with
to
all
say
the
from Dr.
participants?
Saiful or Dr. Kaka? Okay, if there's no question, I think this is the end of our guest lecture. I say very thank you for Dr. Ezatul Azlan for the informative and interesting guest lecture, and also for
Putus. Bu Winda putus. Ya kan Pak Ruswanto?
Bu Winda. Bu Winda suaranya putus, Bu. Okay, Dr. Ezatul.
Ya, Pak.
Last time, we would like to thank and respect for the sharing of your knowledge, and we will share the certificate for you as a
Oh,
speaker.
thank you.
Okay. This is your certificate. Thank you.
Oh, yeah.
Thank you for your
Oh,
sharing
thank you very
in
much.
our lecture.
So, Pak Ruswanto, can we take photo with some of the participants
Okay.
that open the camera?
Okay.
Nanti nak buat macam mana?
Okay. I count this. 1, 2, 3. Okay. I have take photo.
Okay.
Bu Winda. Bu Winda.
Yeah, Pak Rus. Sorry, I have trouble with my signal.
Oh.
Dr. Ezatul Azlan, thank you very much once again. And for all the participants, thank you very much. Mr. Ruswanto also, thank you very much. Okay, I will, hopefully this presentation will be beneficial for everyone. I close this guest lecture. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you very
Terima
much.
kasih Bu Ezatul.
And see you all again.
Thank you,
Semoga
Dr.
berjaya. Ya,
Amin.
semoga berjaya di dalam pelajaran.
Amin.
Good luck for your
Amin.
future undertakings. Good luck for your exam.
Okay. I close the Zoom.
Yeah.